Thanks a lot for the invitation. Thanks to Greg Orr, who kind of organised this visit. It's great to be here. So I'm Chris Jiggins. I was in Ashworth many years ago, so hi to Ashworth people. So I'm going to talk about our work on the black soldier fly, which is familiar to many of you here already. But I thought I'd just start by saying oh, hang on. Here we go. Start by saying that, you know, I'm probably best known for work on these tropical heliconius butterflies, and we still do a bit of work with them in my lab. We're working on the mechanisms of cyanogen toxin production and sequestration from host plants in heliconius. And we also have a big project funded by BBSRC, working on these helicoverpa pests and their invasion of Brazil and hybridization between these two species. So broadly, I'm interested in genomics and hybridization and the evolutionary history of species, and also the link between sort of genotype and phenotype. So understanding how studying genes that are doing interesting things in organisms evolve. So the black soldier fly doesn't really need much introduction here. I think you're probably all familiar with this. This is a video that Tom, my PhD student, Tom, filmed a few years ago of the black soldier fly maggots eating a sandwich. It's always a good thing to show at lunchtime. And so these things are amazing. They grow incredibly quickly. You can feed them all sorts of rubbish, and they can be used, and there's a growing industry around the world using these black soldier fly maggots as a means to convert essentially waste nutrients back and put them back into the food chain. So the maggots themselves, the chickens absolutely love them. They go crazy if you give them live maggots, but you can also produce meal that can be used to feed to animals, or even potentially at some point go into the human food chain. So it's very sustainable, very circular. But currently, BSF is just kind of the species that people bring in the wild, and I'll say a bit more about that in a minute. But there isn't much work on the genetic improvement, apart from, of course, various places around the world, including here in the Roslin. So we can sort of think about three ways, really, by which we could sort of go about genetic improvement or doing a better job of sourcing strains for this growing industry. So one, obviously, would be artificial selection, sort of traditional approach to animal breeding, and we think there's a lot of scope for that. I'll talk a little bit about that, and that's obviously what beta bugs are doing here in Edinburgh. Another would be to harness natural variation. So there's a huge amount of natural variation, which I'm going to talk about in the wild. So we could just pick strains that were best at doing particular jobs, but that would require a lot of phenotypic information. And there are some sort of legislation barriers of sourcing strains from around the world for commercial purposes, particularly around Nagoya. Or finally, we could use genetic modification and do more targeted genetic interventions. So mostly I'm going to talk a bit about the natural variation of black soldier fly to start with, and then a bit about artificial selection. And then at the end, I'm going to talk a bit about better understanding that sort of genotype-phenotype link, particularly with respect to enzymes. So this is a summary of the talk about natural genetic diversity, phenotypic selection on body size, and then understanding enzyme function, which I'll talk about at the end. So firstly, what is this thing that we call black soldier fly, Hermetia illucens? So in collaboration with Christoph Sandrock, who's in Switzerland, he sourced a set of samples from around the world, both sampled from farms, which we called captive sources, and from the wild, so field caught individuals. Of course, the definition of sort of captive versus wild caught is tricky, because they're constantly being brought in from the wild into captive populations, and also presumably then escaping back into the wild. So genetically, it's not clear. These aren't sort of clear categories. But we had some samples from around the world from Christoph, and these were part of a much larger study of microsatellite variation that was published a few years ago by Christoph's group. And we sort of tried to pick samples that were representative of the divergent genetic groups, clusters within Christoph's microsatellite study. So we sequenced about 52, I think, 54 whole genomes using just short read shotgun sequencing to reasonably high coverage. sort of 15x or so. So we got reasonably good, although there was quite a bit of variation in coverage between samples. The sort of take-home message from this is that there's a lot of genetic structure in black soldier fly. So there are two major lineages. And this is whole genome SNP data. The top lineage is mostly the industry strains that are found around the world. So we're calling this the Shepherd strain. That's because a guy called Craig Shepherd in Texas in the late 80s, along with his student Jeff Tomberlin, kind of went out with a bucket and got a bucket full of maggots from a chicken farm. And that founded essentially what is the industry strains that are mostly used around the world. So if you go and get a strain from beta bugs, almost certainly from Beta Origin, from one of the companies here, or also the ones in Asia, they're almost certainly going to be, at least in part, genetically derived from that original founder event in North America. There are some North American samples here, but wild ones, which sort of lie at the base of that strain. And that's why we think there's we don't have many wild samples from Central or North America, but we think that there's sort of a North American lineage within which that industry strain is nested. I mean, that's not really obvious from this figure, but we've kind of looked at other mitochondrial genomes and other sources of data. We think that wild North American and Central American samples are in this lineage, and then everything else around the world is in this other lineage. And this includes some captive populations, as you can see, from Africa, South America, and also wild samples from Europe, Australia, Asia, and we think, and South America, and we think that these are the South American, ancestral South American lineage, which has been spread around the world in association with human dispersal over the last sort of 50, 60 years. So it's probably predates the industrial rearing of these things, and it's probably spread in association with kind of waste and shipping and sort of pre-insect industry. So there are captive samples found in both lineages, but the wild North American lineage piece to be the progenitor of the kind of the industry strain that most people are using. Now, these are really genetically very different. So this is, I slightly take the x-axis with a pinch of salt, but this is a dating that we've done that Tom did on the mitochondrial lineages. You can see the deepest divergence within BSF goes back to nearly 6 or just over 6 million years. And there's another divergence here about 4 million years. This is slightly at odds with another dating using sort of similar methods. And I haven't quite got my head around why we get different dates, but they come up with 2 million years as the oldest divergence within BSF. So it's a sort of ballpark, the confidence intervals around this are pretty big, but whatever, this is kind of on the order of divergence of definitely of quite different butterfly species. If these were heliconius lineages 2 million years apart, we would call them different species. As far as we can tell, and we haven't done the crosses ourselves, but from people who've tried to cross these things, there aren't major incompatibilities between them. So we're not sort of arguing particularly they should be called different species. But whatever, there is an enormous amount of genetic diversity in this thing that we're currently calling BSF. And certainly there's going to be a lot of genetic variation and probably also phenotypic variation that will be useful for the industry.
Chris?
Yeah.
So the Shepherd strain is kind of coming from Texas, and I guess those insects must have come from the south, right?
Yeah.
So kind of crossing Caribbean and Mexico into Texas. Has that been really so long time ago, given according to these divergent dates?
Well, I mean, the Isthmus of Panama was connected about 3 million years ago. So maybe they were, hand-wavy kind of hypothesis. Maybe they were ancestrally in South America. They spread into North America 3 million years ago, and were then somewhat isolated, diverged. And that's what we're looking at. I think this is divergence way before human intervention. Yeah. I think it just has to be. Reviewers of the paper said, could this be because of the industry and inbreeding? But it's not. Right? It's not that. And we think this is likely to have important effects for phenotypic traits for the industry, but the evidence is a little bit shaky at the moment. There was a paper in 2013 that looked at performance of different strains with Jeff Tomberlin, but that really, I think, just showed that there was one strain that was kind of better than the others. The others were probably just a bit inbred, so that was a bit unconvincing. I think more convincing is this more recent paper by Christoph Sandrock showing G by E interactions between strains where you've got poultry feed, food waste, poultry manure, different diets showing some strains doing better on some diets than others. And we've also shown some G by E effects where with 2 or 3 strains on various different diets. And these are actually family reaction norms across diets within a single strain. You can see there's a lot of between family variation within strains on their, in G by E, on their performance in different diets. So this is kind of quite preliminary, but it's sort of the first evidence that actually that genetic variation that we see around the world is going to translate into variation that will be useful for selecting strains that will be good at eating different things in the industry. I don't think that's convincingly been demonstrated yet, but I'm sure it will be the case eventually. Okay. So what are the impacts of the domestication? I mean, I'm calling it domestication, the captive breeding of these things on the genetic diversity in the genome. Well, the first most obvious thing is that there's a reduction in genetic diversity between captive and wild strains. But actually, here you've got on the left, you've got 3 sort of shepherd strains which have reduced diversity compared to these wild strains in the middle, the wild kind of equivalent of them. But actually, these captive populations from South America and Africa, which have presumably been quite recently brought into captivity, actually have a lot of genetic diversity. So some of the, particularly South American captive strains, where I think they brought in South America as the genetic origin of this species, we think will probably be where most of the genetic diversity is found. Some of these captive populations in South America are actually way more genetically diverse than the industry strains that we're using in Europe and Asia. But that's sort of, I mean, what you'd expect, right, is you bring them into the lab, you lose genetic diversity as they have smaller effective population sizes in captive populations. Also, Tom was interested, and I think this is quite preliminary, but we're interested in seeing whether we could find signatures of selection in the genome in the captive strains. And there are certainly some runs of homozygosity, some reductions in diversity in some of these strains that could be indicative of that. So this is the genome sequence strain, and it has a massive run of homozygosity on this chromosome 3, I think it is. No, chromosome 5. But we can't really, with confidence, distinguish between selection and drift really as causes of these regions of lost genetic diversity. A potential route through that is to compare kind of repeated or compare domesticated strains, and this shows actually 3 shepherd strains, and there's some areas where there's kind of significant loss of genetic diversity seen across these strains. But of course, these aren't really independent. This is sort of all the same original event. So this is more really to say, I think this is an exciting area for the future. If we can sample these independently brought into the captivity populations from South America and other parts of the world, then a comparative kind of analysis of putative selective sweeps will potentially be quite powerful for finding those genes that have been under selection in adaptation to the lab. So I think it's going to be quite, I'm not excited about it. I think quite a nice system for looking at the repeatability of evolution, because we've got this kind of artificial selection experiment going on and again around the world in these farms. And then another interesting question from these data are the extent of hybridization and admixture between these lineages. Now, the big caveat to that is that we sort of chose samples that were sort of nesting within these different clades of microsatellite clades that Christoph had. So they weren't really a very random sample if you want to sort of look at the extent to which the clades are mixing up. But we did have a look at admixture between these lineages. Of course, the industry is shipping flies around the world all the time and randomly kind of mixing them up without really any thought of where they're coming from. And they're also then escaping from those facilities and presumably hybridizing with local wild ones. So, I mean, I actually thought the whole thing would be a complete mess, you know, a sort of a genetic soup. And that's clearly not the case. We do find this quite strong genetic structure that I've already shown. And this is a mitonuclear discordance tree. So there's some mitonuclear discordance, there's some lineages here where you can see they're in one clade for mitochondria and the other clade for the nuclear. The sort of nuclear on the left, mitochondria on the right. So there is some discordance between the two, but actually they're pretty concordant with each other, suggesting there isn't a huge amount of admixture between these strains. We had these three sort of industry strain samples that we'd sourced from commercial providers, which kind of fell a little bit intermediate between the two groups. You can see them in grey there, those grey triangles. And so they were sort of candidate group, sort of strains that might be admixed between the two lineages. And so we did a sort of, this is known as an ABBA-BABA test, which is kind of a test originally designed for looking at introgression between humans and Neanderthals. You need an outgroup, and then you're looking for these SNPs, which are going against the phylogeny. So an ABBA SNP is shared between P2 and P3, or a BABA SNP is shared between P1 and P3. So these are relatively rare SNPs in the genome, but if there's introgression between, say, P2 and P3, you expect a bias towards more ABBAs than BABAs. So it's a very kind of simple, intuitive test for introgression. And so we screened across all of the pairs of genomes with this D-Suite software. And you can see a few red boxes here. And in particular, this one up here is this industry strain A, which clearly sharing genetic information or genetic variation with the lineage B strain. So it's an A strain industry lineage with some B strain genetic variant in it. And actually, we can look here. Is this the same? I think this is actually a different example of one of those red boxes on that square, where there's a region that shows high levels of introgression, which is shown on the top panel, and also shows evidence in the wild of a selective sweep. So this is a potential example of something that's introgressed from a captive strain, from the industry captive shepherd strain, into lineage B in the wild in Europe. So, you know, that's genetic variation potentially from captivity out into wild populations. And I would expect that we'll find a lot of this when we the sample sizes here are very small, and the sampling around the globe is very incomplete. So it's just a sort of snapshot, really. So there is some mixing between these lineages. Okay. So this was meant to be me summarizing things so far. Essentially, we have, you know, a lot of genetic diversity, these two clades, which we think perhaps correspond to biogeographic regions of North and South America. But we need better sampling, particularly in South America, to sort of confirm that. We have some evidence for mixing between them, so we don't think they're completely reproductively isolated. I think there is this kind of repeated experiment of domestication, which I think in the future will be a great way of finding the genes that are involved in adapting to industry's conditions. Okay, look away. All right, so next I'm going to talk about selection. And so we're doing something similar actually to what Thomas and Gregor are doing here, but we were really more coming at it from a sort of evolve and resequence approach, where we want to evolve populations and then use that as a way of getting at the genes that are under selection. So the trait we chose was body size. So pupal body size shows less variance than either larval or prepupal body size. So the pupae are presumably showing less environmental variance in their body size. And we have a sort of automated pipeline where we lay out pupae and then photograph them, and those traits of length and width that we get from those images are very strongly pretty well correlated with body size, and they're best correlated actually pupil length with pupil weight is very well correlated. So we can easily phenotype a fairly high throughput pupil length from images, and so that's quite a good trait to select on. And so essentially what we did was we had an industrial sieve. We threw the pupae through a sieve and picked the biggest ones, so we could try and keep the population size large. And we did this in a replicate way in three replicates. So we had a large strain and a control strain going in parallel 3 times. And we did this for 7 generations. So it took less than a year, really, for one PhD student. And we could do phenotyping of selected individuals and the response. So we could get the selected and the response variables for the breeder's equation calculations. And so we could track the response to selection quite well using this kind of imaging of pupae with a pipeline, image pipeline. And so pupil body size is pretty heritable. We got an H squared of 0.36. This is, you can see here the base population in red, the selected individuals in yellow, and then the response, the next generation in red, so from the breeder's equation. And this is the average heritability across kind of all generations and all replicates across the experiment. So there's heritable variation in body size, as you might expect. And we got roughly 16 to 20% response to selection over 7 generations. So this shows just one replicate. You can see that there's the selected individuals in red, the response in the following generation in yellow, and the base population for that generation in blue. So you can see the response is kind of variable between generations. Sometimes it looks more than others. There is a lot of plasticity and there's a lot of environmental variation in this trait. So I think between experiments, between replicates, there's sort of varying amounts of VE, I think, probably. But we got a pretty consistent response in the 3 replicates. So we can select over 7 generations and get 15 to 20% increase in body size. And then another important consideration is, are there trade-offs? So do you select on body size, but they take a lot longer to grow? And actually, not really. So there's a whole bunch of traits. And this shows the mean increase or decrease, the percent change in the control versus the selected averaged across the 3 replicates. So you can see they actually grew faster. They were bigger. The pupil weight was bigger. That was the trait we selected on. They had higher protein content. And the only trait that was a bit negative was female pupil development time, which was slightly slower in the selected lines. So, and then finally, we didn't measure fecundity because yes.
Usually there's always some kind of trade.
Yeah.
And sometimes for the growth or production that we see in other livestock species, there is some kind of reproductive trait.
Right.
On the opposite side.
Yeah.
So do you have any reproductive trait recorded during those 7 generations?
Yeah, so unfortunately not. I mean, we have ovary weight here, which we hope would be a proxy for female fecundity, but we weren't really able to measure female fecundity. So yeah, I mean, it seems hard to imagine there's not some trade-off somewhere. Maria was saying that survival actually trades off. We didn't measure survival in this for these, so maybe there's a survival trade-off. But you would have expected a fecundity trade-off, perhaps, but actually fecundity is very correlated with female size. So if you make bigger females, I'm not sure they'll produce less eggs, but we'll see. Yeah. Anyway, we don't have them anymore. So in retrospect, yes, we should have measured fecundity more carefully, but that's certainly a good question. Yeah. Okay, here's a summary of those things that I've already said. Okay, so then so the phenotypic response to selection was highly repeatable between the replicates. So we did it 3 times. We got very similar response in the selected variable, very similar changes in these other traits. And then we wanted to ask at the genome level whether that was similarly repeatable. So we sequenced nearly 300 individuals, 96 per line. So that was 32 from the base population, 32 from the selected, and 32 from the control. So we've got 3 replicates with the 3 populations sequenced for each replicate. And then we calculated this PBS, population branch statistic. So essentially that's assuming that the difference between the control and the base and the control is what you expect you to drift. And then the extra difference that you see in the selected line are the selected, the loci under selection. So it's sort of a 3-way FST statistic, essentially, this PBS. So it's a way of taking the 3 populations and getting a single statistic out at the end of it as a summary of that. And so candidate regions were 4 times greater than the standard genome-wide standard deviation, which is a sort of arbitrary cutoff. So firstly, just across the genome data, there wasn't strong evidence for inbreeding through the experiment, or particularly that the large selected lines were more inbred than the control. So it's only 7 generations. We kept the population sizes quite high. Replicate 2, we had some CT room issues, and so they underwent a bit of a bottleneck. So we were aware of that when the experiment was going on, and you can see that in the genome data. There was some inbreeding in replicate 2. But 1 and 3 looked pretty good.
Please remind me, what was the population size of the select?
That's a good question. About 1,000. Yeah. So this is then the genome-wide scan for the PBS. And there's actually a remarkable lack of kind of coincidence between the loci in the 3 replicates. So there's a lot of loci that are above our threshold. They're distributed pretty widely across the genome. And there's not very many that are in common across the 3 replicates. Actually, it's not obvious from looking at this plot, but when you zoom in, this region in chromosome 6 is the only one really that has a really consistent effect. Yes?
Are these overlapping windows? Or
that's a very good question. I think they're non-overlapping. And since we've done this, we've done a sort of, because obviously you can have 3 windows in a row, that are really the same peak. And then the other replicate might have this window, and that's sort of, so you have to sort of think about the overlaps. So initially we were just counting overlapping windows, but we've done it in a slightly more sophisticated way by walking out from the peaks, and then count. If you have one that isn't and then one that is significant, then that's probably the same region. So we've kind of I don't have the details in my mind, but we've kind of thought about that, how you calculate the overlaps. And actually, so this chromosome 6 was the only really consistent overlapping region. There are some others that may, but when you zoom in, they're actually not so obviously common. And actually, if you look at genes within the windows, there's only 2 genes across all of this that are in common. And if you look at candidate pathways, sort of this is just a Go term annotation, then the kind of the most significant Go terms are not the same across the 3 replicates. So there's not evidence that they're different genes, but the same pathways. Yes.
So could this be purely because the trait is very polygenic, or could it be due to the 7 generations is not yet enough to fixate specific part?
Yeah, I don't think it's the latter, because we're not necessarily got fixation, right? We've got allele frequency change, and we should be able to detect that, and we should see that even if they're not fixed. I think my interpretation is it's just very polygenic, right? That sort of body size is probably what we expect, I think. But we did find the one, I told you there was one gene, and so that was this chromosome 6 signal. There's some genes that are kind of nearby. There's one that's overlaps, that kind of significant outlier in all 3 replicates. And it turns out to be exactly what you might have predicted, which is the insulin receptor gene. And we've actually done some knockouts. A master's student this year has done some knockouts on the insulin receptor, and sure enough, if you knock out the insulin receptor, they don't grow so much. But this is pupa now of the control versus on the right-hand side, the insulin receptor knockout, which is teeny and quite deformed. So this sort of needs confirming. We haven't actually genotyped these individuals. We don't have very many individuals, but I'm sort of showing you because it's not a big surprise. We know what this does. It's one of the most conserved genes across animals. Insulin pathway is a very conserved growth regulation pathway. So, but I think it's nice that it shows that it's not just noise. These signals of selection. There's some genuine signature of selection in there. Okay, so we can use evolve. Yes, Gregor.
Knocking it out reduces the size.
Yes.
Do you have any idea of what kind of variation there is when the gene is present?
Yeah.
There's
I knew you'd ask that. So we actually did an RNA-seq experiment, expecting it to be upregulated in the large line, and we didn't find a significant difference. Now, it was one developmental stage. We did the RNA-seq from the whole larva, and so it could be that it's locally upregulated, and we just didn't see that in the data. Or the alternative is there's a coding sequence variation that's doing something, and to be honest, we haven't looked to figure that out. So yeah,
clearly it's doing something.
Clearly it's doing something. Yeah. So finally, I got a bit of time. Yep. So finally, my third part of my talk, I'm going to talk about a new collaboration. So this is Danny, who's sitting there at the back, who's sort of initiated this project. And so this is a collaboration with a group in biochemistry in Cambridge led by Florian Hohlfelder, and they work on enzyme function. And really, I think the challenge here is that almost all these genomes we sequenced, we predict the genes, and we annotate the function of the genes based on sequence similarity with Drosophila, probably in most cases is where the evidence comes from. And for an insulin receptor gene, that's a pretty good way of going about things. But for many other things, and particularly for enzymes, small changes in the active site can really change the substrate target. And so annotating genes in that way is inherently flawed. And what we really need is a high throughput way of linking genotype to phenotype for novel genomes. And so that's what we're hoping to do with this microfluidics approach. Essentially, I should put this on just in case you didn't see that. I'll go back, and here we go. These are little droplets going through the machine. These are water in oil, aqueous in oil droplets around the size of sort of an E. coli cell. And these are a way of sort of linking genotype to phenotype. And essentially what we're doing is we've got our gene encapsulated in the droplet. We're expressing the protein within that droplet, and then having some readout of enzymatic activity that allows us to pick out the droplets that contain a gene with a particular enzyme activity. There's various different ways of doing the readout. You can do it with fluorescence or absorbance. Fluorescence is sort of the best, most sensitive, but there are reactions that aren't kind of amenable to a fluorescence readout can be read out in different ways. So what Danny has done is make a cDNA library prepared from the BSF gut. So I should say that this is pretty well-established technique. It's been around for a long time. Some of the patents are expiring already. So it's not totally novel. What's changing, I think, is firstly that there are many more assays that work in this pico droplet format now than there were in the past. But also, I think what we're doing that's novel is doing this with cDNA from eukaryotes. Most of the work that's been done on gene discovery using this approach has been from metagenomes. So basically taking DNA, bacterial DNA from the soil or whatever weird environment you want, and using it for enzyme discovery. And people haven't used it very much for eukaryotes, really, because you need to start with cDNA because of splicing. So you need a full-length transcript to put into your droplet. So that's not rocket science, but it's just something people don't seem to have done very much. So we collect our mRNA, we make a cDNA library, we clone it into E. coli. Oh, there's a nice picture of a BSF gut that Danny has dissected out. So we clone the genes into E. coli, we compartmentalize them into these little droplets. You can incubate them off chip in a tube for a couple of days if necessary, or just for a few hours, depending on the reaction you're working on. And then you have some sort of droplet sorting, where the standard kind of way route for the droplets is down here, but you use an electromagnetic current here to sort of ping off the droplets that have a bit of fluorescence. So you collect off the droplets that have the activity. And so this first assay that Danny ran, she was looking for glucosidases. So these are genes that break down sugar molecules. She started with a rather broad mix of sort of complex carbohydrates, cellulose, starch, a yeast glucan and a lichen glucan. Put the enzyme in with some metals and some potential cofactors. And then we've got this little coupling cascade where if glucose is produced, it triggers a cascade that gives a fluorescence readout. So the beauty of these little coupling assays is that you can take something quite generic like the release of glucose and turn that into a fluorescent readout that you could sort on. And so in the first assay, there were something like 20 positive droplets out of 8 million. So you can screen millions and millions of droplets in a day or in a few hours. Danny took the best ones of those and sequenced them. And then so we mapped them back to the Hermetia genome. And this is one of the candidates that we come up with, which is a myrosinase. So myrosinase is a glucosidase. So it was already annotated as something within this kind of expected function. But myrosinase comes from a sort of plant function involved in breaking down toxins. So kind of annotated from something, related but completely different. And it turns out that this myrosinase actually has undergone recent duplication in Hermetia. This is a tree of soldier fly genomes, and you can see there's a sort of ancient duplication in soldier flies, and then 1, 2, 3 recent duplications within Hermetia. So it's a little gene family that's expanded within the black soldier fly, which is exactly what we might expect if this thing has evolved to be particularly generalist in its functions. Sort of perhaps there's some diversification. So Danny is currently working on cloning and purifying, hopefully eventually, these 5 gene copies, and we can then look at the diversification of function among them. So one of the motivations that we started with in this project is that we thought this very generalist species of enzyme of fly might also have very generalist enzymes. And if you get promiscuous enzymes with general functions, those are really promising candidates for directed evolution. So you can then evolve new functions from kind of weak promiscuous functions of existing enzymes. So our hope was that black soldier fly would be a good source of these promiscuous enzyme functions. But I think it's also going to be a nice way of studying gene duplication and diversification of function. Okay, so that's, and this is this is an AlphaFold prediction of the 5 genes mapped on top of each other. So they're all pretty similar, but 2 of them have this different signal peptide, kind of the yellow and the green sticking out. So 2 of them are a bit different from each other. And there are some differences in their active site conformation, which suggests they might have diversified in function. So that's basically it. Just to summarize, there's this amazing amount of genetic diversity in BSF, and Gregor and I have talked. We're very interested in sort of more of a pan-genomes approach to this diversity and characterizing the structural variation, and that particularly I think the gene copy number variation is going to be really interesting. There's almost certainly lots of genetic variation for industrially relevant traits. I think our evolve and resequence approach to identifying genes, I say it's an effective means, but actually it's been kind of a little bit hard to interpret, I think, because it's so polygenic, this trait. And then the microfluidics is an exciting way of linking gene to function. So finally, just to say thank you. So most of this work was done by the 2 people on the left. Tom above is his PhD work on the genetic diversity and the selection, and Danny on the bottom, who started this microfluidics project. Florian and Matthew are involved in the microfluidics. Miha is our collaborator at Beta Origins, who sort of originally got me into black soldier flies. Shane and Richard were involved in sequencing the genome, and Christoph was our collaborator for the samples, the genetic diversity project. So thank you. Happy to answer questions. Any questions?
Thanks so much. If you have any questions in the room. Yes, Gregor?
Yeah.
You've taken body size because that's very easy, right?
Yeah.
Collect on and
Yeah.
also measure it. And you know, working on black soldier fly now for a couple of years, you probably have ideas maybe which other traits might be of interest. If you would repeat the experiment
Yeah.
again.
So we've had a master student trying to select on fecundity this year. I think we ended up focusing on, you know, the ratio of egg mass to body mass, right? So because of the sort of plasticity of body size, that seemed to be a better trait to select on. So we tried that. It's much harder, right? As you can imagine. It's very impossible to keep the same population sizes that we had with body size.
Did you estimate any heritability for this trait?
Yeah, I don't have an estimate across the whole experiment, but it had a similar heritability to body sizes, just superficially. Yeah. From
I
my
did
body.
not see as much response.
No. He's writing his thesis up at the moment, so I'll tell you in a few weeks. And then I think we talked with Thomas a little bit about, you know, the ability to feed on different foodstuffs with different, you know, composition. I think that's probably, I think to me, those are the two things that are going to be most relevant for the industry. I mean, you have a hub that produces eggs in large quantities, so you want fecundity so you can make as many eggs as you can in a small unit area, and then you send those out to feed on waste products, and you say you need things that are good at churning through whatever waste product it is. So I think those are the things that we've thought of, but yeah, I don't know if that's similar to what you're thinking.
Those will be the typical traits if you take a pig or a poultry
Right.
setting.
Right.
Yeah. You want, you know, good reproduction, good growth,
Yeah.
good conversion.
Yeah.
But I'm wondering, are there any sort of like, you know, monogenic traits? And this is something probably would be very interested in. Have you seen anything interesting? Maybe not commercially very relevant. It depends on their application.
Well, no, because I think we chose the wrong trait for finding monogenic traits. I know that Fira have a strain that apparently can reproduce with almost no light. But that would be quite interesting, with very low levels of light. That's what I've heard, or a rumor. Luke maybe knows more. But so yeah, we don't have other traits that you know, I'm sure there will be, but I don't know what they are.
So for those joining online, feel free to put a message, send us a message if you have a question, or you can also raise your hand. We have the sound on here so we can hear you as well. So any questions from online or any still from the room here? So I was wondering about the results from the beginning, where you were looking at the wild populations and kind of the raised populations, and you didn't seem to see any particular sign of domestication. Is that correct? So I'm kind of wondering if you were to start like a business yourself and start up, would you pick the kind of local populations, or would you go for like a commercially developed population from somewhere else?
It's actually quite hard to bring the wild ones in sometimes to the lab, so they can be hard to establish. They don't necessarily mate. I think there's got to be scope for bringing in new genetic variation in to complement this sort of shepherd strain that everyone's using. So I think probably a bit of both, and I think you know, an exciting area is sort of thinking about hybridization between sort of taking advantage of the amount of genetic diversity there is in the wild, but also not losing the adaptation that was built up over 30 years, 20 years or so, 30 years in the industry strains.
Yeah.
So was there any trick of the trait that the Texas guy used to make them to mate in captivity? It's kind of known that he used that light temperature.
No, I think since then they've got much better at making the lights, you know, so they mate better. But at the time, I don't think they knew that. I think it's just a numbers game. They had a chicken farm where there was just, they came bucketfuls of the things, and if you have enough, you know, a bit like just establishing Drosophila isofemale lines. You know, what they don't tell you is that not all the Drosophila you bring in from the wild get established. So if you have enough numbers, you go through a selection bottleneck at the beginning and pick the ones that survive. Yeah, and it's not impossible, because we have an Ecuadorian collaborator, and he says their strains get inbred over a few years in an industry setting, and they just go out to the wild and bring in a few more, so they can do it kind of on a fairly regular basis.
I once spoke to a lady from Brazil, and she mentioned that she has domesticated them a couple of times.
Right.
She did say it's a bit finicky, but you can do it.
Yeah.
You need to be careful.
Yeah.
Yeah. She didn't really specify what exactly.
Yeah.
Perhaps there is a trick.
Or maybe you just need to keep trying and trying.
Yeah.
It's a numbers game.
Yeah.
Yes, it's quite interesting about what we're saying about bringing in additional genotypes.
Yeah.
We can get a bit deeper. So I remember in sterile insect matter, medflies specifically, I think that was tried a number of times, and as far as they could tell, the introduced genotype
Just took over.
disappeared. No, the introduced ones.
Oh, the introduced. Oh, okay, yeah.
Right, so they just disappeared without trace in a very short period of time, because
Right.
the selection for the matter conditions
Was so strong.
was so strong.
Yeah.
That nothing that came in from out, you know,
Yeah.
nothing came in from outside made it.
That's interesting.
But I think he said there wasn't very much selection, wasn't very much signal of domestication in the first place, if I understood you correctly.
I mean, there are regions that show sort of values of diversity in this shepherd strain relative to the wild ones, which could be selection or drift, and so it's a bit hard to say, I think. I mean, the other thing would be to sort of force hybridization for a few generations, and then hope that you'd sort of get enough recombination that you'd introduce some things that would be beneficial when they went into the mass rearing, sort of enforce the hybridization for a few generations, I guess.
Increased the shepherd strain and the commercial strain, they all seem to be coming from the shepherd lineage.
Yes. So everything, well, I mean, the sample sizes are tiny here.
I know.
So it's very hard to say.
Yeah.
But yes, I think so.
Oh,
It's a very serious one.
question online here.
Are your parents using BSM on their farm? Someone in Ashworth knows that I grew up on a chicken farm. No, and then my parents don't have chickens anymore either. So. I can't see who that is. It just says Ash. Is it Darren? Yes.
All right. So if we don't have any other questions for now, then I'm going to say thank you again, everyone, for joining us. Thank you to Chris for being here today.